Hello, I'm Dan Sweeney from MathSpec Incorporated. In this video, I will go through the theory behind similar partition searching. Similar partition searching is a pattern matching application that basically matches accurate mass fragmentation data to chemical structures. This approach is based on the idea that, in terms of mass spectrometry, small molecules can be viewed as mathematical partitions of the molecular weight. This is an accurate mass MS-MS spectrum of leucine and Keflin in positive ion mode. In this video, I will treat this spectrum as if it were the spectrum of an unknown compound. This video is on the theory behind SPS, but I will first show the results of an SPS search and then go through how we get there. The application uses spectral data in the form of a plain text file of mass intensity pairs. This is the plain text file of the mass intensity pairs of the same spectrum. I've already logged into my Amazon AWS account. To start the searching, I first load up the plain text file. As soon as a new file is added to my user folder, the analysis starts. The results are then placed in the output folder, which might take a few seconds. You can load up as many files at one time as you wish. I'll go to the output folder now. Three hits were obtained here. The leftmost column is the score. The score is based on the number of fragments matched, the intensity of those fragments, and the mass error between the actual fragment masses and theoretical masses. The hyperlinked PubChem IDs are in the center column. Now the dashes in the rightmost column indicate that the mass of the unknown compound was identical to the mass of the hit within the experimental error, of course. If we click on the PubChem ID, we can go into the PubChem website and view the compound with that ID number. The first compound, 3903, is leucine and Keflin. The second hit is also leucine and Keflin. It has a slightly different name in PubChem compound. Now the third compound has a lower score. This compound is leucine and Keflin, where the second amino acid has been, the glycine has been replaced with an alanine. The minus CH2 here, minus a methylene, means that this compound minus a methylene would look a lot like the unknown compound we're trying to identify. So it's similar in structure to leucine and Keflin. Now the middle section is the projected isotope ratios of the hits, and you could compare, if you have isotope data, to the unknown, and the number of synonyms of each of these three compounds. If any of these compounds were pharmaceuticals or pesticides, they'd be classified and they'd appear down here. Before going too far, it's important to explain just what a partition is. This figure illustrates that the fragment ions produced in a mass spectrometer can be viewed as connected pieces of the molecule. Some of these pieces will tend to stay together as groups. For example, the yellow circle may represent one group of atoms that tend to stay together. These groups are usually smaller than the fragments that they're found in, and so they are called subfragments. What we'd like to do is find sets of subfragments that sum up to the molecular weight of the unknown compound. We need to first find the masses of potential subfragments. There are three types of subfragment masses: observed fragment ions, simple mass differences between fragment masses, and finally some differences of three fragment ions. A partition of the mass of the molecular weight would be a set of subfragment masses that sum up to the mass of the whole compound. The really useful partitions are partitions where the combinations of subfragment masses account for masses of multiple fragment ions. Scoring is based on how many fragments can be explained by various combinations of subfragment masses, also taking into account how intense those fragment ions are. Getting back to the unknown spectrum of leucine and Kephalin, I deliberately chose a linear molecule to make this easier to visualize. The computer, in this case our Excel add-in, looks for and scores partitions. These are the top scoring 5 subfragment partitions. The scores are in the leftmost column. Let's look at the first row. I have colored in the subfragments to illustrate how these 5 masses can explain many of the fragment ions. Unlike most mass spectral plots, this one starts at mass 0, so each length is proportional to the weight of the corresponding subfragment. At the top are the fragment assignments in terms of subfragments. It looks somewhat random, but these fragments can be repositioned below, and now you can see how the fragments relate to each other. Redrawing this, at the top are the subfragment masses. The middle are the corresponding parts of the leucine and Kephalin molecule, and the bottom are the elemental formulas that can be derived from the subfragment masses. This is the Excel add-in results for the same data file. I'm switching over to a 4 subfragment partition of leucine and Kephalin, which will be easier to visualize in the next part. The top section shows the subfragments where their length is proportional to their mass. This particular partition cannot explain all of the fragment ions, which is almost always the case. The lower section shows the masses of the subfragments. The subfragment masses are calculated from the data and will therefore have some experimental error. Below that graph are a set of elemental formulas that correspond to the subfragment masses. Quincy Wu showed in 1998 that if one could break a compound into smaller pieces that comprise the whole molecule, that process would considerably limit the number of formulas possible for the whole molecule. Quincy used ms to the 5th, whereas the Excel add-in uses partitioning. Now if we compare the subfragments to the corresponding parts of the leucine and Kephalin molecule, here the yellow part of the molecule, the tyrosine side, has an extra hydrogen relative to the subfragment, while the magenta leucine side has one less hydrogen relative to the corresponding subfragment. Basically, the hydrogens tend to move around during mass spectral fragmentation. So if we break up leucine and Kephalin this way and compare the substructure masses to the subfragment masses, in addition to experimental error, we also have to account for changes in the number of hydrogens. We've been going from an accurate mass MS-MS spectrum to generating partitions. These partitions can give us an idea of what the original molecule looked like. Similar partition searching uses the same principles, but essentially applies partitioning in reverse. Here are 4 ways that we could break up leucine and Kephalin into 4 subgroups. Here the masses of the subgroups are exact masses. These subgroups may differ in mass from subfragments in the number of hydrogens, but also by experimental error. The subgroup masses are stored in a data structure. If groups of subgroups are connected in the molecule, their partial sum can be found in the corresponding column. For example, here the 5th column is subgroup A plus subgroup B, which is 2611113. If subgroup A and B were not connected in the molecule, a 0 would have been placed in that column. The last column here is the PubChem ID number. Leucine and Kephalin can be partitioned about 1,600 different ways into 4 subgroups and partial sums of subgroups. Here 25 of these are shown. In addition to leucine and Kephalin, there are about 245,000 other compounds in the data structure. The next question is, how do we find similar compounds? I have come back to the Excel add-in to illustrate this. In the Excel add-in, there's a yellow button labeled Delta. If a mass is entered into row 22, pressing the Delta button will add or subtract that mass from the cells below. I will enter the mass of a methylene in cell E22. Now if I click on the Delta button, it will apply that change to this subfragment, and also that should change the display up at the top. So basically we've made the white subfragment a little bit longer, and it's only changed the masses of fragments that have the white subfragment in it. Now the 142 subfragment is the one that had the glycine in it, the two glycines in fact, and when we look at a modification of a glycine to an alanine, that's equivalent to adding a methylene group. Now we can also do the reverse. We can put a negative sign in here. And we can subtract from the alanine, we can go back to the glycine. And that's pretty much how the similar searching works. If we find the two corresponding 4 subgroup partitions in the data structure, subtracting a methylene from the orange subgroup of 44568 will shift not only that mass, but all of the blue colored masses that have that orange subgroup in them. To do similarity searching, the subgroup masses can each be sequentially shifted by some delta mass, and then that set of partitions and partial sums can be compared to the fragment masses. Currently, only 7 specific mass shifts are used. These masses are targeted to food and environmental studies. So for example, methionine and keflin would not be found as a similar structure. Thank you for your time.